A very warm good morning to one and all. More greens, more healthy we are. We believe in content in upbringing, up fruitful and gratitude outcome from us to give for our strength is faith of people. Our team has the best skillful members who strive for providing the finest product. Plubit has spiced growth. Tonight it's a task of introducing our founder and our guest lecture. Perseverance, determination, and hard work. Best resemblance are founder N. Venkataraju, MSc in Biotechnology. He is the main reason of this plow and a person who stood by it is Abhinav. It's a great pleasure for me to introduce to our speaker who is expertise in molecular docking and done PhD in biochemistry. It's a task of introducing our guest of honor, and it's a great privilege. I know this is one of the difficult things, but he is a man of his virtue and simplicity. I feel elated to introduce him to everyone in this morning. After all, he is a living inspiration to our young ones who really persevere to learn a great things. Sir, today I introduce you to please deliver your speech. Thank you.
Yeah, hi. Good morning. Nice that you guys are interested in learning docking. Let's see. We'll go through step by step, and I will try to keep it uh, as user friendly as it can be. Let's begin with this. You can ask me doubts any moment, and I would want you to try this docking along with when I'm trying to teach you. Yeah, that takes some time. So there is a handy PPT also. Just go through these steps if you are having doubts. Thank Lloyd for giving me the opportunity to present this workshop. Now, in between, I would like you to turn on your mic and talk to me so that this session could be interactive. The more it is interactive, the better it will be. Coming to the presentation over here, you can see there is a spaceship animation. Okay, there are two spaceships which are coming together. So this procedure of one spaceship coming in contact with another spaceship and making perfect fit into that is also called docking. That's called a spaceship docking. And there are also docking in other terms, which might be related to all the ships, the normal ships at the docks on seashores, or even there are large rivers where you might have seen there are places where ships comes and parks itself, parking that you can relate to that. That thing is also called docking. So from that you can understand what docking actually means. It is like we'll anyways go step by step. So we are not talking about those docking. We are talking about molecular docking over here, but the concept remains the same. So what is molecular docking? First thing that you would need to understand, and why you are learning this uh, should make some sense to you why you are doing this. What is molecular docking? It is a key tool in structural biology and computer assisted drug design. So you can see it's a key tool. It's a tool, something that can be used in structural molecular biology. So you have all studied molecular biology. When it comes to structural molecular biology, we focus on the structures of the macromolecules. Now, in molecular biology, we have all learnt about the macromolecules, the DNA, basically limited to the nucleic acids and proteins mostly. But yeah, so you have learnt about the DNA and the RNA, and a larger section of that is when you come to the translation and you look into the proteins. So proteins, which are the main key players of most of the processes that happens inside your body. Okay. Many of them are also enzymes. So you know that a lot of enzymes play major role in the metabolism. So we need to study about the structure of these molecules, and we try to find out a structure function relationship. Coming though, so that part is can be that this molecular docking can be a big tool in helping us to understand that particular segment. And yeah, of course, it is a computer assisted drug designing. It is definitely an in silico tool. So the computer assisted drug design basically kind of begins with molecular docking. So whenever you're trying to find out a drug, this is going to come handy, very much handy. What is the goal? Why are we doing molecular docking? The goal of ligand protein docking is to predict the predominant binding modes of a ligand with a protein. So this is where we focus when we do not know exactly where the ligand. Now you should understand what a ligand is. Ligand is anything which goes and binds to a protein or an enzyme, for that say. Uh, uh, that is, it could be a substrate, it could be an inhibitor, it could be an activate. So any category of molecule that goes and interacts with the protein is called a ligand. So what we are trying to do with molecular docking, we are trying to predict the predominant binding modes, how a ligand is binding with a protein of the three-dimensional. Now, the protein, of course, you should know the three-dimensional structure of the protein. If you do not know the three-dimensional structure of the protein, you may try to go with ab initio methods and try to simulate the structure, and then try to go for molecular docking. But usually we do not do that. We usually rely with the three-dimensional crystal structures, which are which are definitely much more reliable sources of information. So, yeah, so you know the three-dimensional structures of the protein, and you know which ligand you are trying to find out, uh, which might interact with your protein. So we are trying to predict the predominant binding modes. What does docking achieve, and why do you do it? A successful docking method will search high-dimensional spaces. We have just seen it. It searches for location, so it is going to search high-dimensional space effectively and use a scoring function that correctly ranks candidate documents. So this is something which is very essential. Now, when you do docking, you will have different poses coming up. Different, of course. Once we go through the tutorial, you will understand what these poses are. So it's not just it is going to find only one location. It is going to find different locations at which uh, a ligand is going to bind to the protein. Now, when it is going when it is going to find multiple locations or multiple modes of binding, that is different amino acids at the same location or in different orientation in the same location, it is mandatory that you rank the uh, dockings. That is that must be based on some what do you say, some technical terminologies such as uh, free energy, you know, which is the most important parameter when it comes to ranking. So we check the free energy values, and the least free, I mean the one with the least free energy, that is the lowest value of delta G, is supposed to be the best dock. You know that why it will be, because it has released a lot of energy, so it is going to be much more stable. That's why it gets the first rank over there. Now, second thing, why do you do it? Docking can be used to perform. There are three things that are mentioned here, very essentially that you do. First thing is we do docking to perform virtual screening on large libraries of compounds. When you have very big libraries, when you have so many compounds, it is obviously very difficult to do experimentation in vitro, in the lab, in wet lab conditions, and find out the binding efficiencies, as well as find getting the exact location is obviously a very difficult task. For that, you have to go for crystallization, which is much more expensive and does not really turn out to be successful all the time. So we cannot do a screening of molecules when the library is quite large. So virtual screening on large libraries of compounds is the first thing. Second thing is we rank the results that I just explained you. We rank the results so that we get to know which is a better molecule or which is a better pose, which is a better site where the binding is happening. And third thing, we can propose structural hypothesis of how the ligands inhibits the target. This you can relate to something which is going on now. We all are facing global pandemic, COVID-19, and you know that the whole world is trying to find out the solution to that. So if there are molecules that can bind and inhibit the virus, the viral protein, to attack humans, we can somehow manage to control this the spread of COVID-19, as well as you can get a very successful drug if you have something which is any ligand which is very uh, promising, which is a very promising inhibitor. So we can propose a structural hypothesis of how the ligands inhibit the target using molecular docking. Now, all these things are invaluable in lead optimization. Lead optimization is, of course, finding out the ligand and of finding out the best ligand for our study. Now, how does it work? How does molecular docking work? Molecular docking involves computationally exploring the search space which you're going to see. So we try to explore a search space that is defined by the molecular representation used by the method and ranking candidate solutions to determine the best binding mode. So uh, we are going to explore a certain area, certain space. Now, in the first slide, when you saw the animation, you can understand that it's happening in space. Okay, one spaceship is trying to find uh, how to uh, settle on the other one, dock on the other spaceship. So that could go wrong if it is not properly done. So it can go because it is a space. It's so much of empty area is around that particular another spaceship. So what if it is not going sitting properly over there? What if there is some mismatch over there and docking doesn't happen properly? So these taking that as an imaginary image, you can understand the same thing is done over here. We try to find out, we try to fix a space, search space, where we are going to explore the binding ability of a molecule, of a ligand on the protein. So computationally exploring a search space, and it is defined by the molecular representation. Uh, Molecular representation is the coordinates and the atoms, which are defined in a very different way. I'll try to show it to you. Okay, used by the method. So the method, when we say the method, it can be different methods. There are so many docking tools available, and they have a different approach to do docking. So that's why defined by the method, used by the method, and ranking candidate solution to determine the best finding mode, which you have understood by now, I hope. Ranking. So now let's see what is the workflow, how we go about it. On top, you see there are two things. One is target selection and one is ligand selection. Target is the protein usually, and ligand is the molecule that you're going to bind. So both of these things are at the first level, because without having a proper target and without having a proper ligand, you cannot definitely go for docking. So once you have done that, after you have selected the target and you have selected the ligand, we go for what is called the target preparation and the ligand preparation. Now, we have to prepare both of these species in a particular format. The format will depend upon the software that you're going to use. In this current workshop, we will be focusing on Autodock, which is supposed to be one of the most reliable docking tool. Although it is a freeware, it has been ranked to be the best. So needless to go to Schrodinger or any other. I mean, you can obviously try, but yeah, there's Autodock is a very reliable source, and you can always go ahead with that. So we have to prepare both the protein as well as the ligand molecule in the format, in a specific format. So we prepare them, and then we go for docking. And then once docking is done, you go for evaluating the results. You can go on repeating this as long as you require until you get something which you are looking forward to. Now, all this information on the slides 2 to 5. There is a slide number over here. If you are having any doubts, you please make a note of that.